Over to you, sir.
Good afternoon. During the BioHeal 2025, there were two major incidents, AI-HI war, and after that, the gelatin war. So as an entrepreneur, I am given the responsibility to take everybody out of the campus, a complete paradigm shift, no technical discussion, no lab, no intelligent questions. Listen as a super intelligent, because biomaterials. Can somebody help me, please? Because when we talk about the biomaterials,
Which one, sir?
sorry, nanocellulose. Because when we talk about the biomaterials, most of the talks about the lab scales, academia. So in this particular session for another 20 minutes or half an hour, whatever is the time, we will be away from the labs and we will enter into a plant, industrial plant. As an entrepreneur, what challenges you can see? As a academic student, as a scholars, whatever you are trying to develop here, how it will perform in the real life? Whatever your papers you are publishing, will it be produced in a plant? If it will be produced, what will be the challenges? So what you should focus? I will start with a new question today. Not about what research you are trying to generate here. A new question. Sustainable health. What is sustainable health? Whatever medicines we are providing, they are producing a lot of waste. Still the pharma industry in the red category of the pollution. So there are so many things we need to talk about, we need to address, but only when we come out of the lab. I will talk about a new paradigm, sustainable health. And I will talk about three things. Number one, the crisis. Second, what is the code? And the third one, your. Because until unless we complete the cycle, there is no point of discussing anything. The crisis is current approach to health is unsustainable. Why I'm talking about sustainable health? Because I believe from converting an IITian to an entrepreneur, I believe healing a human being is same as healing the planet. Both are same and both are the same part of the coin. If you are healing the planet, you are healing the human being also. What is the code? Code for this future has been with us for millennia. And where? Hidden in the plain sight. It is the biological and philosophical blueprint of the animal we have lived alongside for centuries, the cow. Here in the various research papers, people are talking about on which animal or which this and that we are doing the clinical trials. So if I say cow, in the session of a biohealth or the advanced health applications, you should not be surprised, but you all are. ये बीच में गाय कहाँ से आ गई? Doesn't match. It will match after some time. Then the cure. It lies in our ability to finally decode it. Decode what? Decode the cow. There are two books in my hand. One is Cowism, the confluence of Sanatan, science, and sustainability. And that is the another one is the Hindi anuvad of it, Gauvad. Sanatan Vigyan or Satata, sustainability ka Sangam. Why I'm particularly talking about the Sanatan and why I'm connecting with the science and how we will attain this sustainability out of it. I will give you a new print, the bovine blueprint. And it will go in three phases: cow science 1.0, 2.0, and 3.0. The cow science 1.0, it is basically a basic simple science of cow, the bovine cattle, where we are taking the Panchagavyas from our ancient knowledge, treating them, reproducing them, validating them, standardizing them in our institutes like IITs and the other reputed institutes. Cow Science 3.0, this is something our advance. In Cow Science 3.0, we will talk about the cow as the core of universal sustainability, the environmental sustainability, a source which is not just addressing one or two sustainability development goals of the UN, but the 10 sustainable development goals of the United Nations. The cow is established as the solution for the water scarcity, water conservation. And that is our cow 3.0. But because it's the platform of BioHeal 2025, I will more focus on the cow science 2.0. That is basically an engine of revolution in the materials. Cow Science 2.0 is talking about the impossible possibilities. For some time ago, people were just ignoring it. It was just a taboo subject. And before I go to the next slide, I will just share one of my experience. A couple of weeks ago, I was giving a lecture, and the event was Gau Mahakumbh. Different. And you know what? Suddenly I came across with one presentation, with one video, and one paper. The date was 1st June 2025. It was from the Berkeley and Edinburgh University. And you know what they were talking about? They were talking about the nanocellulose as a novel biomaterial of future from ruminant bio-waste. And I was very happy. I was very proud, but I was humble as well. Because this call came from the Western countries. And I was so happy because being an IIT Bombay alumni, we had been manufacturing it commercially for last 4 years. Biomaterial for changing healthcare landscape. You know more about it, even more than that. Because every day in various experiments, you are dealing with it. I just want to focus you for this particular one. Nanocellulose, crystalline. We are also producing the fibrous as well. I will not go into the detailed properties because I promised you I will not bring you back to the lab. I will talk about the different aspect of it. Nanocellulose has the different properties. Why I am particularly showing this slide? Because everybody will be having a simple question. Bole baki sab chodo. We know the nanocellulose, we know the properties, we know everything about nanocellulose. But you are extracting it and claiming to be the world's first to extract it from the ruminant bio-waste. Will the properties change? Will it be acceptable? That's why I'm showing you this slide. These are the properties similar with the different sources. My Canadian competitor, they are using the ocean biomass. Canadian competitors, they are using the forest waste, agri waste. This slide is very important. In our research, as a personally funded research, as my company, 4 years of the commercial trials, commercial production, we have done so many tests, so many characterizations, more than 1,000. But this particular slide is very important. You know why? Because this particular slide, this particular test was conducted in IIT Bombay by my guru, my professor, Dr. Jayesh Bilare. And when he produced the report, he just wrote here, the FTIR spectrum indicates that your nanocellulose sample, this word your is very important. Why? Because this was the particular sample which was produced by the ruminant bio-waste. It was basically breaking all the limits that we should remove this subject, a taboo subject. Ruminant bio-waste can be the future source, the major source in the whole world for the nanocellulose. And you should not be hesitant enough to try such type of materials in your labs for your future healthcare applications. So far, you have been developing the test reports, Raman spectroscopy, XRD, and all. First time I am showing you certificate of analysis. First time I am showing you the technical name of this product as a brand, NanoCellX. Even though it is, the source is what? Cow, ruminant waste. The inspiration source is Sanatan. But does it reflect in the brand? No. Does it reflect it anywhere in our technical data sheet? No. Because the technical data sheet is compatible enough, user-friendly enough for our corporates to use. For last 4 years, our clients are all the major companies you can name in country, whether from the paper industry, cement industry, ceramic industry, packaging industry, and of course in the health sector. Again, this slide does not make any sense because you people are wise enough to know the applications of the nanocellulose. मैं तो बनिया आदमी हूँ, धंधे वाला हूँ. This slide is very important because global nanocellulose market is more than $1.2 billion by 2030. And this is in the current scenario where not many applications have been developed. Not many corporates has used it at a large scale, but they are using it. So what is the take for you in this particular institute for the research scholars, for the faculties, for the students that start working on the nanobiomaterials in any form? I particularly push on the nanocellulose. Start developing the applications which any corporate, any pharma giant, any medical sector can use them. रिसर्च वही करना जिससे 4 पैसे कमाए जा सकते हो. पेटेंट वही लेना which you can sell it to somebody like us. People are there to pay for you, pay for the good work, pay for the intelligent and the super intelligent work. Healthcare sector, this is one of the largest segment for the nanocellulose. वो अलग बात है, हमने थोड़ा सा मिथ को तोड़ा है because for us the biggest sector is cement and ceramic. India is producing 200 million metric ton cement per year, which is going to increase. And you know what? If we add 0.5%, I'm talking about the maximum limit. If we add the 0.5% of the nanocellulose in the concrete, it increases the compressive strength by 35%, the flexural strength by 98%. And if only 1% of the total producers are going to use it commercially, what volume we are talking about? We are talking about 10,000 tons per year of the nanocellulose production. And what is the capacity of my plant? Nothing. I make only 1 ton per day. We are the world's third largest. But still, how much is there to go? Just only one sector. And why this particular research is important? Because it's not a research anymore. Because India has the capacity to make the nanocellulose available. मेरे वजह से हो रहा है कि किसी में प्रॉब्लम है? Okay. Why this particular is important in India? Because India has only capacity to be the world leader for the supply of nanocellulose. Why? Number one, availability of the raw material. It's very easily available. everywhere, every part of the country. There is no geographical boundary required for that. Number two, it's a problem for country. आपदा में अवसर देखना है. Every Goshala, every people is working for it. How to find the solution? How to do the systematic disposal management system or the resource recovery of this cow dung or the solid waste. And number three, cow is basically a free of cost bioreactor for us. That is most important. As an IIT and as an academia, maybe I could not see it. That's why I brought you from your lab. Now sit outside and see. Imagine everywhere on the road, the nanoparticles, the nanocellulose is there. And our patent is basically not just about the nanocellulose. Our patent is to extract nanocellulose, carbon nanoparticle, humic acid, fulvic acid, and lignin in a single sequence from the ruminant biowaste. And when we further process the lignin at the lab scale, we have already reached to the sustainable aviation fuel. That means we have completed the entire range of the mineral value chain or the petrochemical value chain just by the cow dung. It's not like there are not challenges. You all are talking about the nanocellulose, you are using it, or maybe another nanoparticles, you are using it. The previous session, there were so many challenges and you were raising the questions. Because you are a student, you can ask any questions. But when you will come to the other side of the table, you will realize nanocellulose is very simple for you. But you know, isko dry karne mein kya ho jata hai? You cannot dry it. When you go to dry the nanocellulose, it requires all your engineering, all your research. Because as you go to dry it, it again agglomerate. And we are not talking about a lab scale. We are talking about a 1 ton per day dry powder. There was no technology available. People are using spray dry, people are using lyophilizers. You can also use them at a lab scale. But what? When you will go to the commercial one, nothing will work. It's useless. You have heard about this supercritical CO2 drying? How many of you heard about it? Anybody practiced? You haven't even practiced in the lab scale. We had done it at a commercial scale. And you know what? As an academia, you will challenge it. You will say, supercritical CO2 drying in an aqueous medium, not possible, not possible. Yes, you are right, it's not possible. So another challenge, how to make it possible? So again, we need to go for another method, solvent exchange. So how to do the solvent exchange? Again, up to what percentage? Message is very simple. There's a long way to travel from here to here. And keep yourself ready for that. Keep your brain open. AI, HI, nothing will work. Just only your super intelligence. Why we are ahead of our competitors? Because we use our Sanatan knowledge, our super intelligence. I see cow as a bioreactor, which is taking the feed. In the phase one, it is working as a multi-pass grinder, free of cost. No infrastructure, no power, no manpower, nothing. Secondly, then do the enzymatic reduction. Third one, whatever it consume, more or less 50%, it converted into pure lignin and pure cellulose and use that cellulose for its own consumption for the energy and whatever it is. Another 50%, it loosens the bonding of lignocellulosic material. And what I get in the return? I get the 100% lignin along with the 50% nanocellulose. We are not worried about the yield. Yield is hardly 10 to 12%. But who cares when your material is coming at free of cost? When your part process is being done at free of cost. And it's not about the yield. The mission is to make the nanocellulose available and affordable for all the advanced health applications which you are going to produce. As a company, just I'm sharing it. By 2025, we already have the commercial high grade facility. GMP medical grade facility, we are working on it. And by 2030, we are planning to be the second largest sustainable cooperative model for the brown gold after milk. Because the cooperative model you have seen, it is only for the milk, and which is the largest one. We are working on a ground level to develop our back-end supply chain and aim to become the second largest cooperative model. What we need to do, what I am doing here, why are you listening me? That is important. The fundamental research to turn this platform into a specific life-saving application, and that is the work of great institutions like you, the wise people like you. It's not my work. I'm done my work. We provide the key. We seek your locks. You create the problems. You create the hypothesis. We will provide you the materials. We will provide you all the possible sources, all the possible funds as institutions. We don't need to wait for our corporate social responsibility to become the corporate. Because the moment you take anything from the cow, it becomes your responsibility to return back. So we are ready. We are working with various institutes on a collaboration basis. Anybody who want to work with the nanocellulose for any health applications or any advanced applications, you are more than welcome. We are working with IIT Bombay. Some of the materials I have already sent to IIT Gandhinagar, Professor Dhanka also. We are associated with Central University of Rajasthan, who have helped us to develop the nano bio fertilizer for the desert application, because it's a biofertilizer, and that too in a nano form. So we are more than happy to associate with people like you. Let us collaborate to build not just a research paper, again I am telling you, but a new sovereign bioeconomy for India. Let IIT Roorkee be the global epicenter for sustainable health innovation, and BioHeal 2025 be the historic platform to shout loud and clear that yes, with our biomaterials, with our Sanatan ancient knowledge, if we recreate them, if we reproduce them in our such institutes, we can change the future. I will just give you, because my another background is from MBA. My master's and second PhD is MBA. So MBA वालों को case study दिए बगैर मजा नहीं आता है. How many of heard about the Chyawanprash? हाथ तो अच्छे से ऊपर कर दो यार. Everybody, I know everybody has heard about Chyawanprash. So what is so special? You know, there was one corporate. Chyawanprash was basically the product, the copyright of our ancient knowledge. One particular corporate picked it up, reproduced, create the labeling, branding, standardization, repack it for the corporate benefits, and that become the brand. डाबर च्यवनप्राश. आज कोई भी च्यवनप्राश लेके आइए, वो डाबर का ही होता है. Second, yoga. Again, it was a service industry. From where? From our Sanatan knowledge. When it was rebranded, when we got a new brand ambassador as our prime minister, it became the product of the world. $1 million, $1 billion का product हो गया. Similarly, we need to come out of our brains. थोड़ा सा खुला सोचना है. This Taoism should be the part of your studies. This particular book, why did I show it? Because it's the encyclopedia of cow. It has all the possible research on the cow. Not just some Goshala, but about the IITs. My friend Samir was here from the Abu Dhabi. King Saud University. They have also done the work on it. There are so many. Tamil Nadu Agriculture University, many universities are working on it. So don't hesitate. Use this material, build something great, and build something which we everybody can proud as an Indian. In the morning session, I heard that AI may we are leading the world, while in most of the things we are behind 20, 25 years from the Western countries. Similar with the nanobiomaterials, we are basically leading the world at this particular moment. And it's your responsibility to take it further and maintain this gap. Let the Western follow us for the nanobiomaterials. In the end, I just give a final call, a choice of legacy for you. To the students in this hall, you will define the next century of science. Will you define it? By refining the old or by building the new? Look at the world's waste and see its potential. To my fellow scientists, the data is clear. And if you want, I can provide you many more. The material is proven, the need is urgent. The greatest discoveries happen at the intersection of fields. Let us meet at that intersection to create a sustainable health, not just the health solutions. Thank you very much. I must say, I'm not quoting anybody, I'm quoting myself. If the 20th century belonged to the synthetic polymers, the 21st belongs to biointelligent materials. Nanocellulose unites sustainability, science, and human health. The bovine blueprint is handed over to you now. The code is cracked. So the question now is not, can we build this future? The question is, will we? Will we? Will we? Will we? Thank you so much. Let's begin. I'm open for any questions. Intelligent.
So it is really wonderful talk. Generally, we scientists in the colleges, university, we focus on the number games. How many paper we are publishing? How many patents? We don't take care about that, or don't we ask ourselves, can it be commercialized or not? We never ask. We just see, oh, it is patented. How many patent? 12 patent. How many commercialized? Zero. How many years the patent existed? 20 years, but no commercialization. This is lot of people they are doing. The number game playing became director, etc., etc. Vice chancellor because number is very important, especially in India. Okay, but now that time is changing, so I am really grateful to Dr. Satyaprakash for opening the eyes of the scientists that how we should focus on our work. We have to ask questions, what we are doing, and why we are doing. Is there any really benefit to the society or not? Maybe okay, he told about commercial aspect, but sometimes the science also people are doing, but that science can be applied for different applications by different groups. So we should think whatever he told, we should believe also and this is truth. Because the other countries, Europe, America, they are focusing how they can get some money from the research. And 100 people, I know 100 people from different area of the discipline, they are working together to single problem, which is not doing. We have a lot of politics. Oh, I will not work with NC Mishra. Oh, Professor P. Anil Kumar is very dangerous person. So better. So this game actually made our country developing country. We are not still developed. because our poor mentality, leg pulling mentality, and not broad thinking on working on some really good challenging problem. So means I am really grateful to Professor Satyaprakash ji.
Thank you, sir.
And uh, one question, okay.
Hello, sir. Here, my name is Khushi. So we also work on cellulose, and we use cellulose for making scaffolds for tissue engineering, basically organoids. So the thing we always encounter with is it is a very stable compound. So dissolving it is a very difficult task, and it is also very expensive and time consuming thing. So like your product which has already been derived from the waste, so it is very difficult to separate out the lignin and cellulose, right? And it is also a very expensive method. So is it like very cost effective, and how are you managing the cost? Because I think we buy sigma 1 and it is 100 gram is around 3400 rupees something. So is your cellulose superior in quality, or is it low in cost? What are the like pros of your cellulose?
As a simple marketing guy, best in the quality, lowest in the price. Rest is all commercial. But anyway, for the institutes, as I told you, because it is not something which I have invented. As I told you, it is reinvented. I just rediscovered from the new lenses of science. That's it. So for the institutes, it's free of cost. Whenever you want to do the research, please make just a simple MOU. What I want against that, number one, in your papers, just acknowledge us. Number two, be confident to mention that the source is from the cow dung. Let the whole world know through your papers. And number three, pure commercial again. If you draw something which can be commercially workable, please join the hands with us. We can do the together patents. We can accommodate in our team, and we can go for the commercial interest. Simple. Any other
Okay,
question?
I think we have now samosa arrived, very hot.
Hello?
So now I request Professor Anil Kumar.
Somebody is asking some question. Yeah, please.
Hello, sir. I had a question actually. Now, really a
Kumar.
great talk, by
Anil
the way.
Kumar का नाम चलेगा ना?
I wanted to ask, what was the biggest limitation you have faced when you're producing these things in the healthcare services, especially in India? Because we know that the national regulatory body, whatever we have, especially for the GMP produced products to be used for clinical trials and all, they face a lot of regulatory limitations in India as compared to that of the global ones. So, what is the biggest limitation you have felt as a producer?
Actually, as I'm not the user of this product, but yes, the users which we are discussing, they face it. But again, as I told you in one of my slide, we don't talk about our origin, we don't talk about anything. What we talk about the certificate of analysis, what we talk about this technical data sheet, what we talk about the MSDS, material safety data sheet. And based on that, we basically pass by all these regulations because our material is passing all the possible tests which are required for the GMP and then the further applications for them. But yes, there are challenges. The major challenge is the clinical trials, the long time. And that basically restrict us. But again, as I told you, we are much ahead than the whole world. We are almost 5 years ahead. The first June 2025 paper from the Edinburgh and the Berkeley University shows that they are still talking on the papers and we are on the commercial production. So we are not worried. I believe we are supporting all the big corporates in the pharma industry for the tissue regeneration, of course for the wound healing, special dressings, precision drug delivery, prosthetics, uh, dental crown, uh, 3D PLA filaments for the different biomedical applications and some of the hydrogels as well. So I believe I am confident about our big corporates working in the health sector, and soon in a couple of years they will be coming with some better products using these materials. I will just share one more information which will again be an eye opener for you. 3 years back, Japanese government, they form a consortium of the 100 big corporates of Japan. And you know what was the task given? To reduce the price of nanocellulose up to $10 per kg by 2030. Clap to कर लो. इससे पता चलता है कि where we are. Japanese, which are the legend in technology, they want 7 years and 5 years from today to reduce the price of nanocellulose up to $10 per kg. हम उससे आधे में आज बना रहे हैं और 4 गुना में बेच रहे हैं. That is the legacy of our Sanatan, the ancient knowledge. That's why इसलिए बोला था कि ताली बजाओ अपने लिए खुद के लिए ही. Thank you so much. If there is any question, or we can wind up.
Okay, I request Professor Anil Kumar to honor Dr. Satyaprakash Vermaji with a prestigious J.C. Bose Memorial Award. Please.
1 second. Maybe first slide. I will appreciate if somebody want to reach me out. You can note down my number 9599. Or you can take the picture of this particular slide. Why to reach out to me? For 2 reasons. Actually, this Bharatiya Gauvadi Samaj, the Public Charitable Trust, the Kavis Society of India, Public Charitable Trust, whatever we earn from this book goes to that particular trust. And that trust is basically working for intervene the small scientific interventions in the gaushalas. Like whatever liquid biofertilizer they make, it has a shelf life of 30 days only. With our small interventions, we increase the shelf life by 9 months. They don't know how to inhibit their fermentation process for the liquid fermentation manure. We provide them the technologies, SOPs for the inhibition so that they can control the pressurizing of that particular container. And now they are able to export these liquid fermented manures. And you know what is the potential of that particular single product? $2.5 billion in the Latin American countries, in the Middle East countries. तो हमारे इस छोटे से intervention से basically gaushalas are able to become the prayogshalas now. And that is our responsibility which we are trying to do. So if anybody want anything to know about this book or buy it book, please more than welcome and spread this knowledge to all the gaushalas, all the people who are looking forward to you for the help. छोटा सा इनिशिएटिव IITs की तरफ से. Thank you so much. I will appreciate our friends also can join us. Yes, please.
For J.C. Bose Memorial Award.
Yes. Please let them also join us.
Please. Gauri Sagar.
Yes, sir. I was interested to ask one question. What solvent exchange that you are doing? Is it aqueous to which salt?
Please.
Sir, please.
This is.
And please.
Oh, sir, come.
Thank you so much.
उस तरफ है.
ठीक है. Please. Yes.
Yeah.
Thank you so much. Congratulations. Congratulations. Thank you. Commercialize the gelatin based by. Yes. So cellulose.
So now,
सर आपकी इजाजत हो तो 1 मिनट लूंगा.
लीजिए.
जबरदस्ती ले रहा हूँ, सॉरी, बिकॉज यहाँ पर जेसी बॉस का नाम लिखा हुआ है. एनीवे ऐसा तो जिंदगी में कुछ किया नहीं है दैट दिस अवार्ड शुड बी गिवन, बट एक इंपॉर्टेंट चीज याद आई. जेसी बॉस बेसिकली evident the life in plants, right? That was his contribution. There are only 3 things in the world which has the power to convert the direct sunlight into the life. One is plant, second is human, and third one is the cow. the indigenous cow Bos indicus, because it has the hump. There are 65 countries in which the cow Bos indicus is there. And only Bos indicus has the capability to convert the direct sun rays into the life. That is called the Surya Nadi. So I don't know how it's connected, but thank you very much.
Biomedical Engineering, IIT BHU. His areas of research interest are cell and tissue engineering, engineered cell therapy for rare genetic disorders, diabetes and cancer, immunomodulation, antifibrotic coating to medical device and implant, biomaterials, 3D bioprinted organs and implants, diabetic wound healing, anticoagulant and antithrombotic agents, nanomedicine, metalloantibiotics, and medical implants. Today, he will be speaking on engineered cell laden hydrogels for rapid repair of diabetic wounds and liver bleeding. Over to you, sir.
Thank you very much. Thank you, the organizer, Ms. Shraddha, Naran Ms. Shraddha, my long-term friend, colleague, mentor. Thank you all the organizer for this wonderful conference. Like, this is one of the platform I have seen, like the talk time, discussion time is like more than the talk time, which is like really unique. And thank you for all the students, like for actively participating to ask the questions. That's like very inspiring to see the new generation is so excited about for like this new gen of research. Thank you again. So lot of tissue regeneration and wound healing we have discussed. So I'll also take in the same journey. And just let me know when like 20-minute finish. You can ask me in between also if time permits. Otherwise, like ask me later. How to change this? Sorry. Follow my hand. Hello. 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 Yeah, I can take. I can take. Anyway, so being a teacher, like I really want to move. If I would have a pointer, it would be great. It's very difficult for me to like stay in a certain situation. So beg a pardon, I'll maybe move here and there. If it is a noise for you, tell me, I'll stand at one place. So I'll take you to the basic science of wound, okay? And then see like how this inspires us to develop a cell-based therapy. Can anyone tell me like why diabetic wound is difficult to heal? Can anyone tell me why diabetic wound is difficult to heal? Yeah. Can anyone tell me why diabetic wound is difficult to heal? High ROS generates. So why like diabetic wound takes or like generates high amount of ROS? Any idea? Because of the high glucose. Exactly. So diabetes is like, as you all know, like diabetes is very persistent and like almost 500 millions of patients nowadays. And 1 out of like 10 patients may experience like their limb amputation or diabetic foot ulcer and all sort of complications. The main culprit is like the high blood glucose. And what it does basically, high blood glucose, what it does or what it changes to the cells. Any idea what this glucose does to the cell? Of course, it does cell death, but it also plays or manipulates these immune cells in few ways. Yes. So what it does is like, you have many of you heard of like this M1 macrophage and M2 macrophage, right? So M1 macrophage is mostly like inflammatory macrophage, and this high sugar try to kind of keep these macrophages in the M1 set, not in the M2 set. In this way, they increase the stress in the body and which cause like further delay in the healing. Apart from that, sugar also cause a lot of oxidative stress and ROS formation. All these cause like a significant challenge in terms of the wound healing. So basically what our lab does, we are by my education, I am an organic chemist, but from my last 13, 14 of year of my research, I'm working in various translational field using like nanomaterials, after that biomaterials, and most currently we are trying to develop various therapies using engineered cells. And many of you will challenge then how, sir, you will keep these cells alive in the body, right? Because when you put these human cells or any allogenic, xenogenic cells inside of the body, they get immune rejected. And that's one of the long-term issue that cell therapy could not push beyond a certain limit. So during my postdoc work, we had developed certain like small molecules. Once we coat with various polymers or devices that could prevent immune rejection. So that was one of our recipe that we could use this cell therapy in a nicer way. And we target various diseases, not only like wound healing, we also target like different rare genetic disorder. For example, ALS, amyotrophic lateral sclerosis, Duchenne muscular dystrophy, type 1 diabetes. So we try to engineer cells, human cells, and put in this immuno-invasive material where the therapeutic cells can live for long. Now coming to this today's talk, what we started with, diabetic wound is really difficult to heal. And many of these wound, not only diabetic wound, all the wound has exactly 4 phases of healing. Anyone knows what are these 4 phases of healing for any type of wounds? It starts with blood clotting. So this is classical physiology, right? So it starts with blood clotting. Second is, of course, the inflammation. Third is the cell proliferation. And fourth is the remodeling. Now, with this persistent high inflammation for diabetic wound, what happens is like, first it takes time, longer time to heal. Second is the remodeling step, this scar formation or the fibrosis or fibroblast deposition. This remodeling step completely gets messed up. And the collagen 3 fiber kind of starts deorganize and deposit randomly, which cause like massive scar formation. And once your wound will form massive scar, it will take long time to further remodel it. So overall, it gets further complicated. And sugar also does one more thing. High sugar can also attract bacteria. So many cases, it also increases infection risk. I don't have to repeat this slide to tell you how important it is to have like research or new technology in this domain. So as I said, these 4 phases are very critical to take care whenever a scientist, an engineer, or a company is trying to plan for certain therapy. If you go or deep dive back to the history of any wound bandages or any wound care product, you'll see like our ancient Indian history, we used to use like a lot of this plant extract or directly used to apply like plants on the wound. And then in the medieval ages also people will use like honey, right? And all these things radically changed after like the theory of Pasteur and of course like this Alexander Fleming penicillin discovery. So from then a lot of new discoveries came. In the 20th century, a lot of hydrogels, biomaterials, this also came to the market. A lot of skin graft came. So this wound research is not very new. Like last more than maybe 200 years, indirectly or directly, many groups, scientists, philanthropists, a lot of people are working on the wound care product. But you will feel amazed to know none of those products actually take care of this all these 4 phases of the wound which are mentioned here. Some of the product only take care of the blood clotting. Some of the product only take care of the cell proliferation. Some may take care of maybe multiple steps, but there are currently no available product in the market that take care of all these 4 stages of healing. And that cause inefficiency in terms of the healing and it further delays. So currently in terms of human, in case of diabetic wound, it takes at least 4 weeks, minimum 3 to 4 weeks to heal the diabetic wound. It can increase further due to complication. So our thought process was, can we develop like a more engineered direct product
maybe a cell based, and can we tackle all these 4 phases? And we have to actively tackle all these 4 phases. So already I discussed the limitation. So this was initially like our thought process, like how we can tackle all the 4 phases, right? So we developed engineered cells. We took the platform as HEK cells, which is a human kidney cells, and it's very good in terms of like transfection efficiency. In terms of tumorigenicity, many was asking, right? So in terms of tumorigenicity, if the phases of the HEK cells are lower, the tumorigenicity occurrence is very low. More than 63 passages, there is a risk, of course. So we try to kind of mitigate the risk by using like very lower passages of the cells. There are other cellular platform also we can use. For example, ARP19 cells, which are the retinal pigment cells. Their tumorigenicity rate is very low. Now, what we did is like we used piggyback vector, which is like a class 2 transposase vector, and it inserts any genetic material in a cut and paste manner instead of copy and paste manner. So it is a permanent kind of integration, genetic integration to the cells. We are not directly delivering the genes to the body. So we have engineered the cells outside to secrete 3 factors. The first one is the factor 8, which takes parts in the intrinsic pathway of clotting, many of you know. The second factor is the IL-10, which has its well famous, well known anti-inflammatory mechanism. And the third one is VGF, one of the hallmark of like protein that influences the angiogenesis of the cell proliferation. So in this way, not all these 3 factors is inserted in the same cell. The VGF and factor 8 was inserted in one cell, and then the IL-10 was inserted separately into another cell. The cells were encapsulated in the alginate, which is a well classical kind of biomaterials delivery system, is biocompatible, FDA approved, and this is well cross-linkable with barium or calcium. Why it is beneficial to have like well ionic cross-linkable? Because they are well stable for longer time. So we have checked in my previous work that cell encapsulated alginate cross-link with barium can stay in the body over a year without any degradation. That's also very important because there is a very low risk of cell escape. So I'll not discuss too much about in terms of piggyback vector and system, but if you want to know more, you can discuss with me. It just one thing, it uses one like helper plasmid. So as we kind of control the helper plasmid or wash it away after the transfection, the risk of further gene hopping or risk of like further escape of this gene and causing like tumorigenicity or genetic changes in the body almost is negligible or nil. So that is one way we try to avoid any side effect. I'll not bombard you with too much of result. Those are all conventional experiment you all know. It's just to support, can we cure the wounds, diabetic wounds in rapid time, right? What we did, we first kind of transfected the cells with VGF and factor 8. We confirmed that if the cells secrete VGF or factor 8 or not by ELISA. It confirmed that yes, it can secrete. And then we did some scratch assay, wound scratch assay, to show like, can this combination of the therapy heal or not, heal the cells or not. You can see like it rapidly kind of narrowed down the wounds. After that, we did some like pro-angiogenesis assay in chicken egg, which is basically like a model of like to do the CAM assay, where you can see like within like 24 hours, there is a multiple fold changes of in terms of the vessel diameter and all. After that, we check that our cells, are they stable inside of the capsules? Are they alive? Are they proliferative? Like with conventional live dead assay and cell proliferation assay, you see like even after 7 days, the cells are like alive. And just remember, this therapy we can anyway manipulate because these are external therapy. So we can kind of decide like maybe after 5 days, we can give one more dose. So up to 7 days, there was like almost 50% of the cells were alive. All cells were proliferative. There was no sign of significant toxicity. Then same way, we engineered the cells for IL-10, same transfection method with lipid-based lipofectamine. And then we confirm there is like multiple high fold of IL-10 secretion from those cells that confirm like the perfect transfection. Once the transfection is done, we utilize like a hydrogen peroxide based like stress model in the cell to see like if the IL-10 production can reduce the oxidative stress or not, using like very conventional DCFD assay. And you can see like the green fluorescence was quenched once we use like IL-10 based therapy. It is like kind of reduce the oxidative stress. Same way in combination, we check the scratching assay, and in a combination group, it was very highly effective. So in terms of VGF, in terms of IL-10, the in vitro results support our hypothesis. Now we wanted to see like if this factor 8 which is getting produced from the cell can clot the blood in rapid time or not. What we did for that, why this thing is coming? So if you, the figure is little bit cut, but if you can see there, we have utilized human blood and then we incubated with the serum media. So we lyophilized the serum media which had this factor 8 and we incubated the human blood with the factor 8, and compared to the control which takes about like 40 to 50 seconds to clot, our material clotted almost like within 25 to 30 seconds. So there is a rapid kind of enhancement in the clotting time. Further, we did APTT and PT assay, which is like conventional assay to see if the clotting is going through intrinsic pathway or extrinsic pathway. So basically, the APTT, we saw some. So you can see like both PT and APTT was done, and it is like it showed response in the APTT pathway. That means like the factor 8, as usual, as everyone knows, it clotted the human blood through the intrinsic pathway. We wanted to further check if this clotting can be also shown in like in a mouse or rat model. For that, we created like a liver bleeding model. So basically, we pricked the liver, we created an incision, and then with the alginate hydrogel, we mixed this lyophilized powder of like serum powder, which had this factor 8. You can see the blood clotting time has severely reduced. The amount of total blood loss also severely reduced. Okay. This also confirms that factor 8 is perfectly working to reduce the bleeding in the animal model. Then once all this in vitro along with the factor 8 activity through the ex vivo using human blood was like proved, we wanted to test if we can create a diabetic wound model and can cause like a very rapid healing. As I said, like even in the mouse or rat, depending of the wound size, of course, it may take up to like 20 to 25 days to heal the wound. So our goal was like, can we heal the diabetic wound with a like 10 millimeter size at least within like less than 2 weeks? So in that way, we created like STZ based diabetic model. This is not a genetic diabetic model, chemical based diabetic model. And as you can see, like within 13 days, the lead group, which had both IL-10 as well as the VGA plus factor 8, almost like 95 to 98%, it healed the diabetic wound. We did some mechanistic study. We found like it has like CD31 like proliferative cells that are like classical cells for like angiogenesis and blood vessel formation. We also check this macrophage polarity. If the M1 macrophage, M1 or M2 macrophage is actually overexpressed, we found through the CD163 staining that M2 anti-inflammatory macrophages were recruited near the area. That also supports like there is a potential of this type of therapy to reduce the inflammation when the diabetic condition is there. We did some other staining also. We did ELISA also to see like if certain inflammatory cytokines are reduced or not. For example, TNF alpha, IFN gamma, those were also reduced in the animal model. Further, people would like to know always like after the therapy, what is the fate of the capsules, right? Are they intact or are they broken or not? Although the wound therapy was only for 13 days and it was like externally done. So there was no risk that the capsules may break completely inside of the body. But just to satisfy reviewer concern, what we did is like we also implanted the capsules in the intraperitoneal area because we also had this liver model. And we kept them for 1 month. We explanted the capsules, almost all the capsules we implanted. There was no like fibrotic deposition or any inflammatory marker deposition on the surface of the capsules. Why? Just because of this effect of anti-inflammation by the IL-10. And we didn't see any sign of like bleeding, any sign of like abnormal tumorigenesis. Although also you can understand that 28 days is very short. So our goal is also to maybe keep these capsules for 1 year and to see like if there is any abnormality or any other sort of issues. So we just recently, like 2 weeks before only, this work got published in Chemical Engineering Journal. Molloy, the first author, is here, and maybe you can ask more questions to him because he will present his poster. And I thank like all funding agencies. DBT actually sponsored our work. So we are highly grateful to DBT, Government of India, to fund our work. All other funding agencies, thank you, like all PhD students. Some of my PhD students are here. So you can go through like their poster and find out what other type of work we do. Thank you again, and thank you again the organizers. So yeah, so this is our happy lab. We do a lot of work, hard work. Along with that, we go for a lot of enjoyment. So we are planning to go for this Char Dham next year. So hopefully I'll take Naranda with me for the Char Dham trip. Okay, thank you. Thank you for your patience. Please, if you have any question, you can ask. Thank you.
I have a doubt.
Yeah.
You mentioned that alginate has good degradation rate.
Not good degradation rate. Yeah, please continue.
But you achieve like you told 1 year of. So isn't it degraded? It doesn't it degrade by
So
then?
you can manipulate a degradation by the extent of cross-linking, right? So barium and so if you make the capsules with only barium, they are very highly cross-linked. So you can keep them for a year. But if you can manipulate the cross-linking with calcium or maybe a mixture of like barium and calcium, you can curb it down maybe like within a month. For our case, this is a short-term therapy. This is external therapy, so we don't care that much about degradation. But we have done, for example, in 23, I have an HRBME where we have implanted like beta islet cells for diabetic reversal, and that was for a long-term therapy. So we use same alginate with barium chloride for 1 year. So you can manipulate it. It's a very like useful polymer.
Great. Thanks. Thanks a lot for such a wonderful talk. My question is on the in vivo studies. Generally speaking, when you have to make a diabetic wound rat or mice model, you introduce the STZ, and then you have to wait for a couple of weeks, even
Yes.
more,
Yes.
for first of all confirming that the mouse model or the rat model that you're getting is actually diabetic, and that there are like certain regulations which say that it has to be about certain deciliters per whatever per ml of the blood, and then and only then you will categorize that as a diabetic rat model. But here you are injecting the you're looking at the therapeutic potential only after I think a couple of days or 5 days.
7 days. After
7
7
days.
days, because it was directly intravenously injected with a high dose. So as you put like a very high dose in rat with IV, so it can generate within like 6 to 7 days.
But you don't do the characterization before
We did.
starting
We
the
did.
therapy.
It is already established. Like we had like few control set where we generated it, waited for 2-3 days, and the glucose level was like persistently high and it was stable. So all those characterization was just to ensure that the model is stable enough.
Oh, just last comment.
Yeah, sure.
I'm sorry for this comment. It's a devil's advocate comment. When you are, for example, working, I saw a lot of data, especially the in vitro data as well as the in vivo data. I did not understand the premise of the experiment that you did on the bleeding experiment on liver. That is of the least concerns to any
That's
diabetic
of
patient.
the least
So,
concern.
but why would you then go and do that experiment in the first place?
Because we don't have like a bleeding setup with us, right? So we
But you don't
have
need to.
this.
One does not need to study bleeding in a diabetic patient.
It
Or
doesn't
is
need
it? I don't
It
know.
doesn't need to for like a conventional diabetic patient. But for example, you have diabetes and then you encounter an injury or accident.
Oh,
What about that? Right? So in terms of a conventional system, it may not be required for a small wounds and ulcer. But same, because 500 million of diabetic patient is huge, and many of them are daily basis experiencing like
Yeah, yeah.
proper injury which is causing bleeding, maybe like a liver disease. And many of these actual liver bleeding, why we also choose, many of the nowadays tumor formation, disease formation is happening in the intraperitoneal area. And even though there is huge expertise of the doctors, still during doing surgeries, there are like minute liver like resections and other injury. So that was the back of our mind, and we kind of kept that
Oh,
model.
great, great, great. Thanks a lot.
So
Thank you.
thank you for asking that.
Thank you.
Thank you.
Hello. Sir, you told about, sir, yeah, actually we are also working on this aspect. So you told regarding some, I will repeat the same one from diabetic wound you are working. When after 7 days you are telling us a high dose of streptozone, it is diabetic in nature. Then how much time you given for the acclimatization to establish that it is diabetic?
Initially, like for example,
Because I've done number of experiments on diabetic, I only just wound and diabetic. Even
there is two type of model,
and
right?
I've seen problems in rabbits also. There are strong regeneration power. So it takes me so much time to establish that.
Which animal you use?
We have used all the rat, mice, rabbit, everything.
Which mice? Which strain of mice?
Is it normal mice?
No. No.
Bulbs
Bulbs is quite resistant in terms of diabetic generation, and it reverses back the diabetes. So you have to use C57 pro-fibrotic mice. That is highly kind of like they get this type of immune disturbance, and the model stays there for long.
But in rats, how much time you take for the
Rat,
acclimatization?
it can take one single dose, high dose. It will be like 7 to 8 days. The model will generate and is persistently stable. And if you want to generate for like like 5
For
or
example,
6
today
dose,
you have got the results that it is diabetic in nature.
it is diabetic.
With some glucose
We
estimation.
measured the blood glucose over time.
Then after how much time you get better established that it is continuing in that nature?
It is continuing till, so we are not curing diabetes. So at the end point also the animals were having diabetes, right? So there is nothing like we are curing diabetes. So diabetes is persistent for till the study endpoint.
Hello, sir. Sir. It's a very nice talk. Sir, I have some questions. Like you have transfected VGF and factor 8 in some cells and in IL-10 in some cells. So how you screen that which cells contain these factors and these cells?
Just by ELISA. You can do PCR also, but by ELISA you can just take the protein productions.
Okay. So have you checked the release profile of these factors from the
Yes,
alginate
we checked till
hydrogel?
7 days they were releasing consistently.
So
The release study was pretty consistent.
So up to only 7 days or
No,
like
you
20?
can check up to 14 days also.
So it is a like a cumulative release only.
It is accumulative release, or you can also remove the media from the plate and see like continuous release also.
Okay, so and in mice experiment, so you use this hydrogel in like what lyophilized form or like it is in the
No lyophilized form. It is like with capsules with live cells.
capsules
Yes.
and,
And we
okay.
try to add like some saline in between. And
Okay.
therapy, how it works is like they'll release the protein, right? So basically these capsules will and cells will stay viable for like 1 to 2 days. And after that, anyway, the mice and rats scratches their body and leave the capsules out. Okay. Again, after day 4 or day 5, you have to give one more dose. So that's one issue with like rodents, like they more often like kind of scratches their body even you apply certain band-aids. So you have to multiple time you have to give the dose.
So normal diabetic wound healing, it requires like 4 to 5 weeks.
Yes.
So it reduces around 2 weeks. Like with this, if we put
Within
if we
13
use
days,
this,
within 22
hydrogen.
weeks, yes.
And okay, thank you.
Hydrogel can you use? We are changing different pH.
Yeah, mostly pH responsive hydrogels are well and good in terms of like this drug delivery in the tumorigenic area. But I think we can use like pH and stress condition in the diabetes and maybe activate like further releasing of the drug.
Okay.
Because if we measure, I have not measured the pH for diabetic condition, but I would believe like it might be little lower than 7.4. Although I don't have any idea, I have not measured it.
Okay, sir.
Okay, thank you everyone.
Very good.
So I request with Professor M.K. Sir. Hello. Later. No, he is also student doing PhD under you, I think. He is student, I know. But once I started, not allowed. So I request Professor M.K. Gupta to honor Professor Sudeep Mukherjee with the prestigious J.C. Bose Memorial Award. Sir, please. Now our next speaker. क्या नाम है? राजेंद्र. Now our next speaker is Professor Rajendra Kumar.
Dr. Rajendra Kumar Singh is an assistant professor from Institute of Tissue Regeneration Engineering, Bangkok University, South Korea. His research interests include nanobiomaterials, biomaterial engineering, ceramics, and tissue engineering. Today, he will be speaking about smart push and pull nanotherapeutics for osteoarthritis, targeted drug delivery with dual scavenging of ROS and
This one.
cell-free DNA. Over to you, sir.
Your mic.
Mic नीचे.
Niche.
Hello. Good evening, everyone. Thank you, Professor Misa and Professor Bhisam. They invited talk for this conference. I'm working in Bangkok University, South Korea, under Institutes of Tissue Regeneration Engineering. This is our institute's name, I train. You can search in Google very easily. So this is our building. So we are working, this is the 6th floor for InVio. 5th floor for the material work and cell work, and here is our sitting place. And 4th floor is kind of the instruments, all facility here in 4th floor. So we have very big area. We are working around more than 80 people together. We have many collaboration. So today I am going to present smart push and pull nanotherapeutic for the osteoarthritis targeted drug delivery with dual scavenging of ROS and cell-free DNA. I know everyone know the ROS scavenging, but here is the together with the cell-free DNA scavenging. This is the new things in this project. So we know that osteoarthritis is a very complex disease. They have many complications. When have the osteoarthritis, so we have pain, we cannot walk. This is more dangerous than the cancer. Because we cannot walk, just you can realize COVID, 2 years, we are in home. So without walking, we cannot do anything. So osteoarthritis is created by the body weight, age, and some accidents. And suppose we have any kind of the chronic disease, also they create. So in osteoarthritis, they involve the inflammation. Why we feel the pain? Why osteoarthritis is the degrade? Because of the inflammatory cell. This inflammatory cell can eat any kind of the cell. So they kill the chondrocyte cells, and then after that we feel pain, and we cannot walk. So how we can control the inflammatory cells? So when we have healthy chondrocytes, so here have no porosity. जब आप चिकन खाते हो तो चिकन का लेग पीस कितना टाइट होता है, cartilage. Very difficult to penetrate any kind of the particles, any kind of the drug. Otherwise, doctor can inject inflammatory drug near the knee and very easy to repair. But this is impossible to penetrate any kind of the drugs. But when we have osteoarthritis, so they make the porous. And porosity will be increased, and but retention is very high. Retention means suppose we can inject any kind of the drug nanoparticles, so they will make very fast clearance. So we can see here is a different kind of the size of the nanoparticle and different cationic polymer, cationic charge and neutral charge. So when we inject the nanoparticles, so they will make very fast clearance through the blood vessels. But when had the positive charge of the particle and ultra small particle, so we can see here when have the size is small, so they will penetrate inside the cartilage. So how they penetrate inside the cartilage? This is very important questions. So when cartilage is degraded, so they have the kind of the porous structures, and this protoglycan is a highly negative charge, and this protoglycan connected with highly cationic polymers. So when cationic polymer is means combined with by the electrostatic interaction, so nanoparticle or any kind of the cationic particles slowly they will penetrate inside the inside the cartilage. So when they penetrate, so their efficiency will be increased, and we can deliver the drug maximum to maximum inside the cartilage. So in this project, we choose the PAMAM. PAMAM is a very highly positive charge. This is G3 structure of the PAMAM, and one molecule have 32 amine groups. So this is very highly, suppose in any kind of the particle we can functionalize with the PAMAM. So here is a highly positive charge. So this positive charge connected with the protoglycan, and they will penetrate, and efficiency will be increased. What is cell-free DNA? Cell, when has cell is dead, means otherwise cell have secretion, apoptosis, necrotosis, they release some kind of the DNA. This DNA is different, similar to means delivery DNA. So this fragment DNA size is 150 to 100 BP. Just excuse me. So this cell-free DNA, how they increase the inflammations. So this cell-free DNA, they follow the DAM kind of the pathway. They interact with the cartilage cell, and they follow kind of the DAM pathway. So this DAM pathway is a kind of the receptor, toll-like receptor TRL9. So they are connected with the cartilage cell by the TRL9 receptors. So this cell-free DNA, they increase the inflammations. So when how cationic nanoparticle quench the cell-free DNA. So when nanoparticle functionalize with any kind of the cationic highly positive charge, so cell-free DNA is a kind of the highly negative. So they will bind by the electrostatic interaction, and then after that they condense and neutralize, and they fragments. So definitely in your mind, normal DNA we can load and deliver, but here cell-free DNA, here I am trying to the neutralize and condense, because of the both DNA structure is different. And when they make the complex and neutralize, so inflammation is decreased. So we know that the ROS is a two kind of the ROS, up regulation ROS and down regulation ROS. So when have the up regulation ROS, so they kill the cancer cell, and they kill also bacteria cell. But when have the down regulation ROS, so they decrease the inflammation. So in case of the down regulation ROS, people are using antioxidant particle. Antioxidant particle, they decrease the inflammations. So in this project, I choose the ceria oxide nanoparticle. Ceria oxide nanoparticle is a kind of the autocatalytic activity, because ceria have the 3+ and 4+. So depend on the environment, they will change 3+ to 4+. This is a kind of the cycle. So here is the objective of the project for the dual screening, ROS and cfDNA, and drug delivery and penetration. So total had the 4 objectives in this project. So first we developed the mesoporous ceria. This is very simple methods and productive methods. We can develop a large amount of the products. Here developed by the autoclave methods. And then after that, functionalize with succinic acid, so they will create the carboxylated group. Carboxylated group is highly negative charge. So PAMAM is the positive charge, so this is very easy to functionalize. So we can see the major porous cilia nanoparticle size is less than 80 nanometers, and we can see in TM image they will show the major porous. But what concentration we can use the PAMAM? So here we did different weight percentage of the PAMAM. So 40 weight percent PAMAM have the higher Zeta potential. So here we use the whole project 40 weight percent. So we can see when we functionalize the PAMAM, so we can see very thin layer. Here is the form. Then after that, we characterize the major cilia. So we can see here when we functionalize the PAMAM, so they have the near the 40 millivolt. This is very highly charged, and also we confirm by the XPS data. So we can see when functionalized with the G3, they had the nitrogen peak because of the amine groups, and how much PAMAM is coated with the major porous cilia. So we observed, we did by the TGA, so 3.6% weight percent. But we observed that the XRD and BT measurement, means pore size and surface area, there have no any big difference between after functionalization and before functionalization. Major porous area is a kind of the multi-enzymatic property. Why they will show the multi-enzymatic property? Because of they had the 3 plus and 4 plus ions. So the ratio of the 3 plus and 4 plus is very important for the ROS. So here we can see the before functionalization and after functionalization, there had no any big difference. Because when functionalized with any kind of the polymer, so they have no, people are reported they have no any kind of the ROS difference before and after. So here we did TMBSA, SOD, DPPH, free radicals ion, means peroxide screening, and how this is hydroxyl radical screening. Then after that, we try to load the drugs. So dexamethasone is a kind of the inflammatory drugs, and this is a highly negative charge. So actually here we use not dexamethasone, dexamethasone phosphate. So our particle is the positive charge. So this is highly negative charge. So very easy to load. So 1 milligram around 50 microgram loaded. Then after that, we check the release with a different pH, because when we have the inflammation near the environment, so there have pH is in little acidic environment. So this is the region we check different pH. So then after we also loaded the FITC, because we try to check the efficiency of the stability of the particle through the FITC. So we can see the FITC is loaded with the nanoparticles. So for the DNA screening, we use the Calf DNA for the binding. So we observed that the different concentration of the DNA, they load 40 microgram per milligram. And then after that, we also try to the heparin. Heparin is a highly negative charge, and DNA is also highly negative charge. So we mix together, and we try to check the efficiency of the nanoparticles. So when have near the 100 microgram per ml of the heparin, so DNA binding efficiency around 40% is decreased. But this is also very high. So then after that, we check the TRL9 cell. So we treated with the CPG-ODN. CPG-ODN is a kind of the drug. So when we treat it, so they will increase the inflammations. So this is the positive control. So when we treated the nanoparticles without and with functionalized, so with functionalized we can see when concentration change, so inflammation is decreased. But without G3, so almost similar to the positive charge. And also we check the another cell, raw cell also they increase the inflammation. So we treated with CPG-ODN, so we can see the inflammation increase. When we treated with the G3 and G3 particles, so we can see the inflammation is decreased. So it means they will quench the ROS. So we mimic the first we culture the raw cell and then treated with the CpG so inflammation is increased. Then we treated the nanoparticles. So this nanoparticle, they scavenge the cfDNA, and this condition media transfer to the chondrocyte. So we check by the PCR matrix synthesis, matrix degradation, and inflammation. So we can see the when we treated the G3 particle, so inflammation is decreased. And MMP, this is the matrix degradation. Also, we can see that this is decreased. So then after that, we try to the ex in vivo model. So we cut the cartilage 6 mm and thickness is around 3 mm, and then we create the osteoarthritis by the enzymatic degradation, and then we treated the nanoparticles without G3 and with G3. So and then after that by the ELISA we confirm that the CFDNA, MMP13, TNF alpha, and IL6. So we can see the cell-free DNA is decreased when particle is functionalized with G3. But we also observe when without functionalized also CFDNA is decreased, because these nanoparticle participate kind of the two kind of the role, ROS scrunging and DNA scrunging. This is the reason without functionalized also CFDNA is decreased, and we can see the inflammation also decrease when we functionalize with G3. So again we check by the under ROS condition. So chondrocyte cell treated with H2O2. So first we check the intracellular ROS. So when we cell treated with H2O2, so intracellular ROS increase, but when we treated with nanoparticle with different concentration, so ROS is decreased because of the antioxidant particles. So we also check the cell viability. So cell viability is increase with H2O2 concentration, and then after that we under H2O2 condition we did the PCR data, pro-inflammatory, antioxidant, matrix synthesis, and matrix degradation. So we can see the matrix degradation when particle have treated with G3 and without G3, so they have the decrease the matrix degradation, and inflammation also decrease. So after that, we mimic the in vivo models similar to the in vivo model. So we culture the microphage cell and treated with cfDNA, and then after that this soup transfer to the chondrocyte cell and then treated with nanoparticle, and then we did the RNA sequencing. So we can see we can see the RNA. This is heat map. So with the color we can see the differences, and then after that we did the geo analysis, DNA repair, and ROS process. So in DNA 30 genes and ROS process around 15 genes. So and then after that we confirm the geo analysis. So DNA repair. So DNA repair means cell will be stay under cell stay under with mimicking of the under oxidative stress. So we observe that they will show the DNA repairs. So this is ex in vivo model to confirm the penetration and how much nanoparticle penetrate inside the cartilage. So we cut 6 mm and 3 mm thickness, and then after that enzymatic degradation to create the OA model, and then the nanoparticle treated. So this is by the homological lysis, and then TMBAC to calculate the nanoparticle concentration. This is a kind of the reverse process, and the cross section, how much particle penetrate inside the cartilage. So we can see here when in OA model, so in G3 case is a higher number of the nanoparticle penetrate inside the cartilage, and we also observe that the penetration depth. So in case of the G3, they have around 150 micrometer they have to penetrate. So nanoparticles functionalize with FITC, and then we injected in OA model, and after 3 days we sacrifice. So we also check by the fluorescence imaging. So we can see here they will show the rainbow colors. So it means particle stay near the knee. So why particle is stay? I already explained, because of the
protoglycan is the negative charge and our particle is the positive charge. So the nanoparticles slowly they penetrate inside the cartilage. This is the reason particles stay inside the cartilage. So this is the last in vivo model. So here we choose the TMJ model. This is very near the here. So this is a TMJ model, very small models. So TMJ model created by the CFA. So when this is the healthy one. So when we treated with CFA, so we can see they show the kind of the porous. It means cartilage is decreased. So when we treated with the CNP, G3 and dexamethasone, so we can see this is start to the recover. So this in vivo we did after 7 days with sacrifice. And then after that we did the H staining and safrin. So we can see they have to regenerate the cartilage. And also that we confirm by the collagen, MMP13 and inflammation. So we observe that we can see the inflammation is decreased in case of the CMP, G3 plus dexamethasone. So the conclusion, this is very easy to develop the cationic antioxidant mesoporous nanoparticles. And these cationic nanoparticles rapidly penetrate because of the highly positive charge. So this mesoporous area we can deliver. And here is a two kind of the squench. CFDNA, they will decrease the inflammation, and ROS squenching also, they will decrease the inflammation. So two kind of the squenching. So this is also cartilage targeted delivery. And this is the things. So this paper have also cover images. So this is Hewang Kim. He's our director, and generally he's a vice director. So our group and Nancy she did the in vivo model. Thank you very much.
Any question? Oh.
Just
Intelligent student.
Thank you, Professor Rajendra.
Mm.
Excellent talk, excellent talk. And so this CDM, like for example, did you also check like about like any macrophage polarization or any inflammatory signals? Does it also play in terms of the osteoarthritis treatment, or just based on the oxidative?
Just based because when have osteoarthritis, so this environment is a kind of the oxidative stress environments. So this is the reason we check by the osteoarthritis. This is very harsh condition. Oxidative stress is very harsh condition. This is the reason we check.
So you didn't characterize any macrophage here, right?
No.
Okay. Okay.
Because this is very well-known material. So
No, CDM is like Professor Sudipta Shil from US is working since long with CDM.
yeah, I know Sudipta. Yeah.
This is an excellent addition to the group of CDM. Nice work.
Yes.
40 degrees Celsius.
Yeah. Autoclave, yeah.
How was it done? I mean,
Just Syria. I think I used the Syria nitrate autoclave methods, right?
Yeah. No, but in which machine? I mean,
Autoclave, high pressure and stainless steel autoclave. This is made by the stainless steel. Just you
that
can.
I know, autoclave I know, but 24 hours.
Yeah, 24 hours. Yeah.
So the chamber can hold water up to 24
Yes,
hours.
yeah, because this is made by the stainless steel. Yeah. We are doing not 24 hours, 3 days. Yeah. Because this is the crystal growth. Why people are using autoclave? Because they had the high temperature and high pressure.
No,
so
okay,
yeah.
Sir, that I got. But 24 hours continuous or in between?
Continuous. No, continuous. This is the first time prepared the mesoporous area, because people have published nano size around, because I have already published nano size around 20 nanometers. This is the first time to develop the mesoporous area. This is similar to the bioglass, mesoporous bioglass. So we can use.
So I think there are no more questions. And let us congratulate Mr. Rajendra Kumar C. Wilson Great Memorial Award. I request Mr. Chapu Chap, please present this award to Dr. Rajendra Kumar C.
I want to inform my master also from IIT Roorkee, 2002 and 2000. From physics department. I am physics student, but doing cell biologist.
Mr. Chapu was also from physics. So now I request Mr. Mukesh Dhanka from IIT Gandhinagar present a keynote address.
A little about Dr. Mukesh Dhanka. He's an assistant professor from the Department of Biological Sciences and Engineering, IIT Gandhinagar. His research interests are biomaterials, drug delivery, bio-nanotechnology, orthopedic applications, regenerative medicine, photothermal therapy, cancer, and autograph therapy. Over to you, sir.
Change from here. Hello, good evening, all of you. I think most of the things related to biomaterials have been discussed in wound healing, tissue engineering, drug delivery, and so on. So what I'm going to do, I will just briefly talk about endoscopically injectable hydrogel. And before I discuss about this technology, I would like to tell you that who we are and what exactly we do. So, like she already mentioned, I'm assistant professor at IIT Gandhinagar in Biological Sciences and Engineering Department. We have a group of people from different backgrounds. You can see here we have genetic engineer, we have plant biotechnologist, we have DNA nanotechnologist, we have people who are working in something ultrasound detection and so on. So we have all kind of people. This is the all are professor in biological science engineering department. This is the beautiful campus located at bank of Sabarmati. You all are most welcome to visit sometime. You can just let me know if you want to visit. We will take care of everything. So the research that I do is not like basic research. There are two types of research. Basic research, you just discover something, publish, and done. Rather, this is something translational research. So what we do, by training, I'm biomedical engineer. We develop biomaterials and drug delivery technology platforms which can be used specifically precisely to manage the significant, or you can say the unmet clinical needs. So we develop these materials, we develop these technologies in the lab, biomedical technology in the lab. So you know this is the pathway how you do or how you conduct the research. You first hypothesize something, what technology you can develop. You test in the in vitro condition, figure out whether it is working in the way you want or not. Then to be able to take it further, you conduct the preclinical trials. You can scale up the biomedical device, whether it is scalability is very, very important. Once you figure out, yes, you can scale up the material, yes, you can scale up the technology for the large people for the large population, then you can go ahead. And then we test these technology in the small animals. After testing in the cell culture and the and mammalian cells, we test in the rodents models. We use all kind of models, small animals and large animals. Then we evaluate the toxicity, and then we go ahead with the clinical trials and further translation. So it is again translational research, not something basic research that we do. Uh, this is my lab, biomaterials and drug delivery at IIT Gandhinagar. And these are the problems that we are working on. So again, like people in the conference, they discuss about the polymers, they discuss about the carbohydrate, proteins. Satya Prakash ji, he discuss about nanocellulose. So we use all these polymers, all these materials, we figure out their properties, we have the library of these polymers, small molecules, we figure out their properties. Even we can use the polyphenols as well because they have the bioactivity. We figure out their properties, on the basis of property, we figure out their application. For example, if you want to develop something artificial synovial fluid, which polymer you prefer? You prefer the hyaluronic acid because it has the properties that completely mimics or matches to your synovial fluid. If you want to develop something skin flap, you prefer to use collagen, you prefer to use hyaluronic acid, gags, and so on. So if you want to develop some nanoparticles, rather using some other synthetic polymer, you can use polyphenol self-assembly. They can deposit layer by layer through pi interaction. These are physical interaction, and in between you can entrap your chemical drug that you want to deliver at the localized site or at the precisely or the target site. So this is what we are doing. We are developing all these nanomaterials, microparticles, hydrogels, composite, to be able to manage these unmet, or you can say the significant clinical problems. I think I don't have to show the data related to wound. It is very significant problem across the world. Sudeep already showed that how many people are suffering from this one, but we are also working in this area. So as I mentioned that my area is overlapping with several of people who already presented, so we are open to collaborate. So, but at IIT Gandhinagar, we have established mainly 3 areas. We have developed the technologies which can manage the arthritis, septic arthritis, rheumatoid arthritis. There are different types of arthritis, osteoarthritis. I think one person already discussed about that. That was very fantastic presentation. So what we are doing basically, we are developing artificial synovial fluid which can support the movement of the joint. At the same time, it can elute out the drug in the controlled, or you can say rather in the on-demand manner. So you don't have to give the multiple injection in the joint because you can't take the drugs for a long period of time through the oral route. It's going to create a lot of toxicity in your body. So you have to be very careful. You have to be cautious while delivering the drug. So this is a rather localized drug delivery system, long-term drug. Up until now, we don't have any system which can deliver the drug in the joint cavity more than 3 months. Everything will clear out from the joint. So this is the big challenge in the biomedical engineering. Another that we are developing the biomaterials, nanomaterials, smart hydrogels, specifically for management of the wound and something like bone infections. And third problem that we are working on cancer, like you can develop the drug release system, but rather we have developed something hydrogel material, the platform technology, which can help to precisely remove the tumors from the body, especially from the GI tract, gastrointestinal tract, that you eat, absorb, digest, everything. It is so important organ in your body. We have the funding from following organizations, agencies, and we are thankful to them. So this is the overview of the work. I'm going to just discuss that little bit, tell you these are the technologies already been developed. We have filed the patent for several technologies. We have published several papers. If you want to visit, just visit our lab, or we can meet and discuss more about that. But right now in this talk, I just want to focus on one technology, that is endoscopically injectable hydrogel, that we have already developed, published, conducted the large animal studies, and we are waiting for clinical trials. And soon we will start that. So the polyp, hydrogel for the polyp removal. It is precancerous tumor. What is polyp? It is precancerous tumor. It can occur, so there are different stages of the cancer. One is very like benign stage. It is not cancerous, but over the period of time, it can convert into the cancer, hyperplasia, and then cancerous in situ cancer formation. If you can go through the screening in your GIT, endoscopic screening, 100% you can cure this disease at very early stage. So screening or diagnosis is so important, so crucial if you really want to completely treat this condition. So this is very, very important tool, endoscopy. So these are something like abnormal polyps, small tumors in the occurs in the GI tract. There are several reasons: genetics, food that we eat, pesticides, so many things. You can read so many things, but all these are factors which are associated for the occurring of this polyp in the GI tract. So, and you can see the prevalence. It is third most common cancer among all the cancer and second most deadly cancer. So it is not something. Recently, I saw several newspapers in that it is saying that the prevalence of this colon cancer and GIT, it is increasing in India. you can relate this scenario that the food, processed food, and all we were consuming. So we have to exercise, laziness, everything. All these are contributing. So we have to be very, very careful while taking care of our health. So the endoscopy is being used primarily in the clinics. It is being used to clear out, to remove these tumors. And that is the most and crucial tool that we have right now that can immediately at early stage remove the tumor from the body. However, how does it do that? Uh, there are two methods which are mainly being used to remove the tumor. So one is endoscopy mucosal resection, and another is dissection. In the resection, this is the wall of the GI tract, you just try to scrape it off and you can just remove. Another is just you take the cut around the polyp, you just put the endoscopy, screen it, where is the polyp, figure out, just then cut it off. However, you must be thinking, oh, this is working fine. What is the need of doing the research in this area? There are certain limitations, and limitations are following. First, if you are, while doing, these surgeons, they are very quick. They want to complete the work as soon as possible. They try to scrape, they are trying to scrape it off, there are high chances, major risk that they can leave some cells over there. And that can come back again. It can form the polyp again. And you think about India, people once are done, they think, okay, I'm done. I don't have the disease. Why should I go again for the screening? They don't go and it can come back. Second, while you are trying to remove the polyp through the dissecting, there are high chances that you can create a large wound or you can damage the wall. So just look at the wall of your GI tract. You have mucosa, submucosa, and muscular propria. That is the muscular tissue. So if you just damage, it can create a big problem for the clinician. It is difficult to manage. So for precisely removing these tumors in the targeted manner, what doctors they are doing in the clinics, they are using some solution which can be injected beneath the polyp and remove the tumor. Otherwise, it is very difficult to remove. So these are the just water, NaCl, or some just glucose, dextrose, all these things, they are mixed. Why they are using this solution primarily in India? Even in India, we still have been using this solution because the length of the endoscopy, you put the catheter, that is the tube, small tube, you put it is 35 centimeter long, and then you have tiny needle, 26 gauge needle. So I think it is very difficult to inject any other material, only water or something you can inject. It's easy to inject. That's why they are using. But what the problem they are getting, as soon as they inject there is a polyp, as soon as they inject beneath the polyp in the submucosal layer to be able to protrude the polyp so that they can cut it off, as soon as they inject, it diffuses out because it is water, a small molecule, nothing else is there. So it is going to diffuse it out. They don't get enough time to remove. It doesn't form a stable, long-lasting cushion. So to be able to get the depot, long-lasting cushion under the polyp, so that these doctors, they can nicely, precisely remove that polyp in a proper way. That is the problem that we are facing. So in already products are there, polymeric hydrogels are there, polyoxymer-based hydrogels are there, cellulose, some other hyaluronic acid have been explored. While going through these hydrogels, which are already commercialized, I figure out they have certain limitations. One, first of all, we have all worked with the polymers. They have inherent viscosity. So it is very difficult. Injectability is the crucial here. You have tiny needle and long catheter. So they have inherent viscosity. So it is difficult to inject, first. Second, hyaluronic acid and some other polymers, they have sort of property like when you inject, they can stimulate the cancer cells and form the tumor rapidly than in normal rate. So these are the problems, and they don't have any smart properties, like they cannot stop the bleeding after removal of the tumor. So what we thought, that can we develop something indigenous technology that we can commercialize and help have the societal impact. So what we did, we discussed with several doctors in the Ahmedabad. We went to the door to the hospitals and discussed with the surgeons and endoscopy people what they are using. All they said that we are still using the saline. We don't have LVU, that product available in India. However, some companies are buying, they can provide you, but we don't, as such, we are not using any other materials, no hydrogels, just we are using the saline. Then we went ahead and we asked, are you really facing this problem like rapid diffusion, difficulty to remove? They said, yes, we are facing, but as such, we don't have any other solution. So what we should do? So we discussed, can we develop any hydrogel that can easily inject it through the endoscopic catheter and remain over there, provide the long-lasting cushion? Would you use it? They said, yeah, we will use it. If something is there, we will try it. We are happy to try. So that's what we did. We developed submucosal self-assembled hydrogel. Again, what is self-assembled hydrogel? What is amphiphile? I'm going to just give you a brief idea about the amphiphile. This is very small molecule, just like your fatty acid. You know that hydrophobic part, you have stearic acid in that, and then glycerol. So it is diglycerol of stearic acid. Glycerol is hydrophilic part, stearic acid is hydrophilic part. You put this small molecule, and it is very small molecule, it's not polymer. It doesn't have inherent viscosity. So you just put it into the water, heat it up, and you will get the self-assembled hydrogel. Why? Because the hydrophobic part of this molecule is going to interact another hydrophobic part of the another molecule. This in the same category. So they will, through self-assembly, hydrophobic, they create the nanofibrous structure. Okay, and they just self-assemble, put them together, and you get the self-assembled hydrogel. When you apply something temperature, when you increase the temperature, or when you apply the shear force, injection force, it is going to disassemble the fibers. It will become the liquid, will be able to inject easily through the catheter, and right after injecting into at the localized site, it will again self-assemble from the gel. So gel, sol, gel. That is the method that we use. So this is plant-derived DGMS molecule, small molecule, highly biocompatible, doesn't cause any problem, no immune reaction, nothing, no hemolysis, nothing. So we just use water, solubilize it, and then develop the hydrogel. Now, one more problem. What if you can encapsulate some dye to be able to visualize where is the gel injected, and to be able to visualize the tumor where is exactly, so that you can simply cut the tumor only, not without damaging other part of the intestine. So that is very crucial. Now people are developing some other advanced hydrogel which can manage these wounds in the intestine, because managing the wound is crucial challenge. So we encapsulate the dye. We also encapsulate the hemostatic molecule that can just clot the blood over there. So this is whole idea. It is biodegradable, easily injected through the catheter, and this is the material that we have developed, prevent the bleeding and so on. So we just developed this hydrogel through self-assembly, and then we checked the injectability. First, we checked, just use the normal needle, 26 gauge to 20 gauge and all. We figure out that we can inject easily without applying so much force. Even we check the injectability to the 3D bioprinter needle, that is tiny again, and you can see the image over there. It formed very 3D bioprinted kind of material that can be used for some tissue regeneration and other things. But yes, up until now, we have just explored this in drug delivery and tumor removal. So here in the same image, you can see these are the nanofibers. You can see, we just visualize that nanofibers were there, and we bought this catheter from the market. We figure out that when you put and inject it in the in vitro condition, it is coming out through the same process. Applied shear force, gel becomes liquid, flowable liquid, and again, right after injection, it regain its structure like three-dimensional, or you can say the nanofibrous structure. We wanted to check whether applying the shear force really it is getting into the liquid stage or low viscosity stage, and then again regaining its gel state. So we just, you can see the rheological studies, viscosity came down and again come back. Then we just give me 2 minutes. Just so you can see the diffusion of dye, and when you put into the solvent, it is retaining into the gel. And then we wanted to check the degradation. It remains in the life. So specifically in the intestine, you have the lipases which can degrade the molecule. So right after injection, lipases comes and degrade, and it can come out through the body through in the fecal material. And it can actually clot the blood, because we have hemostasis molecule, chemical molecule encapsulated into this one. So we tested in vitro condition, actually in ex vivo, we bought got intestine, we put the hydrogel in that one, and you can see the stable cushion. So this is the cushionite in comparison to saline, dextrose, all it remained over there and you can see the height was really remarkable to achieve. So we tested in vitro, like in animals, it was completely biocompatible, not causing any tissue damage at the localized site, not causing any triglyceride level up, and not causing any further, also not causing any immunological reactions. So we tested this technology in the animal, in large animals, pig model, and we figure out that in comparison to saline, it can form a stable, long-lasting cushion, and you can remove the tumor nicely. And we did that in vivo conditions. So basically, this is the material that we are going to translate. We are obviously going for clinical trials, easy to prepare. We can scale it up. We can make in liters. There is no issue of that. It doesn't have viscosity, high viscosity and all. We can store at room temperature in clinics or medical shops. So we are looking for collaboration with the industry partners and other labs which can help us to, I already discussed with Dr. Mukesh Gupta ji, is the director of ICMR, how we can translate that. However, we have the plans, and so we are going to that. So we have published in small, we designed this cover page for the journal. We got the patent application. It has been covered by several news agencies, and we got the invitation from IIT Madras to translate as well. So thank you. First of all, thank you to all of you for being here. And I would like to thank Sabir. He's my collaborator. We have published so many papers, and we are continuing with that collaboration. We have very strong and established collaboration with Muzammil here, Shokin Muzammil from Kashmir. So all are my collaborators, students. I would like to thank everyone, and thank you again.
So no questions. Later you can question because he crossed the time. If time crossed, no question. Okay.
Yes.
But you can discuss with him later. So very wonderful talk and lot of work, hardworking person. Okay. Maybe he will achieve a lot in near future. And I request Professor Shabir Hassan
One pic.
to honor Professor Mukesh Dhanka with prestigious Wilson Great Batch Memorial Award.
Thanks a lot.
Not smiling, not smiling, not smiling. How to spoil kara degi na? हाँ, spoil करा. नहीं है ये serious.
One more. I know, that's why it was there.
Okay, smile.
Great, thank you very much.
So our next speaker is Professor Gauri Shankar, Rai Mahapatra from Oldia Institute of Technology.
A little about Dr. Gauri Shankar. He's an associate professor from Department of Applied Sciences, Haldia Institute of Technology. His area of expertise lies in organometallic chemistry and heterocyclic carbene complexes, applications of catalysis and bioactivity, such as anti-carcinogenicity, antibacterial and anti-tuberculosis study, and conceptual density functional therapy. Over to you, sir.
Good evening to all the audience here, especially to the convener, Professor Chao, and my beloved elder brother, Mishroda. We belongs to same municipal area, same district, same city, and I am humbled enough to get the invitation from IIT and representing my institute. We are in a older institute in Eastern India region, the first ever private institute because before the Monipal Institute also. So in our group, we generally previously worked on the organometallic chemistry, synthetic, and its characterization and application to different aspects. But due to the lack of infrastructure, now we have moved to the computation. Because in computation, with some computers and some softwares and some other things, you can at least generate some data and publish something. And sometimes it helps to the synthetic chemist for their near future to synthesize it. And what we have done, we are lucky enough that one group has published our what we will show the result today and very recently. So in our case, I always start all my research presentation with the father of Indian chemical science, Acharya P.C. Ray. I will come back to these slides at the end of this presentation. But one thing I must note to you, this is Indian Chemical Society, which was established in 1924. It is 101 years is running. Though today I am representing Indian Photobiology Society, don't mix it, two things there. Indian Chemical Society is the first chemical society in India, not only in India, in Asia also. And that time there was 4 society, all are instituted in Calcutta. First with Asiatic Society, then Indian Association of Cultivation of Science, then Indian Science Congress Association, and the last one in 1924 by Acharya P.C. Ray. And he is probably the first person for whom today we are here. Because that time the science was studied in a general mode, and that time in most of the university, 3 university was very popular: University of Madras, University of Bombay, and University of Calcutta. Everywhere the science degree was given in BA in science, MA in science. So BSc, MSc came later on. But the first person who came back to India with some of his students, one of his very legendary students is Bhatnagar, the founder of CSIR. With his help, that time he instituted the Indian Chemical Society. He had a dream that it will be very similar to Royal Society of Chemistry, but it had not happens due to all of us, because we have not that legacy or not transferred that potential to the new generation. So today, whatever I am talking in front of you, I dedicate to that the 100 years of legacy of Indian Chemical Society. And I will request all of you to be the associated in the Indian Chemical Society, because not only for my lecture, because Acharya P.C. Ray was the first person who developed some leaders in science in our country, who instituted one by one institute. It is from Meghnad Saha, from Satyanarayana Bose, and Bhatnagar himself, and the first IIT, which was in IIT Kharagpur, the first director was his student. So he spread all his students throughout the India, not in India, even in Pakistan, even in Bangladesh. And from there, the scientific culture grown in our country. So that's why we called him the father of Indian chemistry. But there is a date in National Science Day, but till date there is no National Chemistry Day. So I think from IIT Roorkee, we can at least take this message that 2nd August should be celebrated as the National Chemical Day. Anyway, so sorry. So I just skip this title. So we are trying to model some compound which can adsorb the hydrogen as a fuel. So hydrogen as a fuel to adsorb or to to carry forward from one side to another side. Only one system is the liquefaction in the cylinder. And the major problem is that 2 major concepts. One we studied in class 7, that hydrogen, when it's burned, there is no flame. So we cannot understand that hydrogen is burning somewhere. So the hydrogen car, which is nowadays being established and running, if there is any flame occurs, so nobody can find out that it is flame already occurred. So people will burn inside. Nobody can identify it. Only the heat we can understand. Second thing is that if you want to, reserve the hydrogen in a cylinder, then you need a huge pressure, and even it should be in cryogenic temperature. So, but this technology is already being used, and already Reliance has, given a huge amount of money for this research. And recently Adani Group has spent a huge amount of money in this research. But 2 aspects is there, don't mix it. So Government of India is also sponsoring a huge amount of money to the CSIR Sikri in Kerala, Karaikudi, to split, the seawater to hydrogen and oxygen. So this is the technology which we have the Israel only. So we are connecting to that indigenous technology. But we are on the second aspect, that how we can absorb the hydrogen and can deliver to the industry. So there are so many, scientists have tried, especially they have tried in organometallic systems. This year, the MOF has got the Nobel Prize. Many people have tried in MOF also. But in every case, there are some certain failures. So, but what is the beauty of this hydrogen, as a fuel? Because the prime cause is that if you burn hydrogen, then the effluent will be water. That is the water vapor. This is the first thing. So in green chemistry concept, whatever the technology will come, this is the first and end technology if you can convert hydrogen as a fuel. Because nothing is there which will convert something to water. And water is the main focus of the green chemistry. And if it established within few years, though it is not established yet, then it will be another heavier process. Because probably nearly 70 years we are there, or 100 years more, the nitrogen plus hydrogen to ammonia, there is no second method till date. We have studied in our HS books or class 10 books that this is this of the iron as a dust to be used as a catalyst for the surface area, huge surface area. But the problem is not like that. It is the iron 1, iron 2 to iron 3, the one electron transfer, the electron voltage is very minimum, and that's why the hydrogen is going to be effective role. But the sweetest thing is that in our body every day we are converting nitrogen and hydrogen to ammonia, but outside our body we cannot, because there are some proteins inside our body which can bind with iron and can do that. So this is another types of typical problem which are facing to hydrogen as a storage. Okay, so now come to the real problem that is everybody we know. We have written so many essays and paragraphs. What is the bad effect of greenhouse gases and like that? So from this chart, you can see in the last 20 years or nearly 1950 to 2022, I have taken the data. What amount of different kinds of fuel we have used and what amount of CO2 we have produced. So the basic advantage of hydrogen, I have already told, this is the most abundant, most lightest fuel. So to carry forward, to carry from one place to another place, the weight problem is gone. So these are the several types of work which has been done. That is the compressed gas method, which I have already explained. Cold cryo-compressed, which I have already explained. Liquid hydrogen you can produce. And by the MOF, by the boron nitride, by the lanthanides, by the sodium aluminum hydride, by the sodium boride. So many compounds people have studied, but it is a major challenge. So why it is so major challenge? Because in 2010, DOE, US, because you always try to follow US, right? So US, they govern, now the Trump was also creating the same problem. He's saying that what I am saying, everybody will learn to that. So they say that the DOE, if something is hydrogen storage material, it must have two property. One is the absorption energy, and second thing is the weight percentage, how much hydrogen it is capturing. And they have given a data that 6.0 percentage at 2010, and later on they have developed it to 9.0 2015, that if this kind of amount being absorbed in a particular compound or a surface, then we can say it is a good absorption material. And obviously the favorable enthalpy, we all know chemistry student, so del G will be negative, that I know, but the process should be cost effective. That is another part. So if we go to that, then what will happen? That it will be clean energy without pollution, most abundant element in the universe, lightest fuel, richest in energy per unit mass. This is the most important part, and can be stored easily, can be produced by water, direct conversion into thermal, mechanical and electrical energy. Sorry, sir. Sir. So these are different process. We are only going to our part. So if you design a compound, which aspect, because we are dealing with the computation, which aspect you will first target? First is that whether the hydrogen is absorbed in the molecular phase or atomic phase. If it is in atomic phase, then your research is gone. Nobody will touch your paper or nobody will come to your result. So obviously hydrogen will be in molecular form. So in our case, what we have taken as a strategy, right? So it may not be a good strategy. big scientist will come with other strategy, but it is also established because we have published nearly 35 papers on this. So the first what we targeted that the hydrogen in a molecular form, the nearly the bond length of hydrogen is 0.74 to 0.76. Huh? How many times? 10 minutes. 15 minutes. So I have taken only 5 minutes. At least come to the result. Okay, so coming to the result then. Okay, so what we have targeted, now I am coming to that, that we have taken the copper as a metal and mixed with the organometallics, and copper organometallics we have prepared, and where we have studied the hydrogen storage capacity. And to store that, we have taken the simple chemicals which are available in the lab, and we mixed with copper, and several computational methods we have taken. As the time has limitation, so I am not going to that. And these are the different calculation method that you can follow in my paper. And these are the different equations that we have used. Now I am coming to the results. So these are the molecules which you have taken, and all the molecules can take at least 10 hydrogen if 2 copper is binded there. And if it binds, what will happen? The organometallics, organic compounds have the aromatic capacity, whether the aromatic capacity is gone or it is there, we calculate it. If the aromaticity is sustained, then the stability of the compound is there. Then we calculate the, adsorption energy. So there is a specific region. If the adsorption energy, average adsorption energy is 0.8 to 0.8 electron volt per hydrogen, that will cover your or satisfy your result. So that we get. So I am not going to that. Already the time has gone. And second part is that how much distance is your hydrogen from your molecule. So it cannot go beyond 3 angstrom, and we calculated all the molecules, it is within the 3 angstrom. And the what is the type of interaction. So it is not chemi-absorption, not physi-absorption, because if it is chemi-absorption, then compound will form. If it is physi-absorption, from surface it will go. So it is within that, so it is a quasi-absorption molecules. And we have studied with different parameters, and these are the summaries of the results, and we have published in some papers. And fortunately, those came to some of the cover image of some journals, and these are my students who have done this. 3 of them have already awarded PhD. This work was I have showed today, it is the work of Abhishek. I am thankful to him, and thank to the audience for giving me the time, and thank to the organizer for inviting me, and thank you. And already the time has gone, so according to the rule of Mr. Da, if somebody has any questions, you can ask me in the lunch hour, dinner hour.
So other time, yes.
Yes.
So no question hour. Okay. And now it is wonderful work. So let us congratulate Professor Gauri Shankar Roy Mahapatra with Meghnad Saha Memorial Award. I request Professor Anil Kumar, please. Professor Chao, please join Professor Anil Kumar. And Professor Jeng, you also please come. You are sitting for a long time. Please come, join. Yeah, please, up. Okay. So now I have some important announcement. We have every day gala breakfast, gala lunch, gala dinner. So there is no special gala dinner or etc. So today is also we have gala dinner, but dinner will be at KIH. Breakfast and lunch only here, adjacent to L1.